welcome you all for the 8th class, which is we are into the second week. So, before I proceed, in the first week we have a little bit of a spillover into the second week, and in the second week there will be a spillover into the third week. That way we will be able to understand the course in a much broader perspective. So, today look at the slides. So, this is the aspect we will be dealing with, need for economic census to meet the emerging threat of biosecurity, and we are supposed to cover the case study of filoviruses like Ebola and Marburg, and then the anthrax, and then the botulinum toxin and insulin. So, to start off with Ebola, so there is a book I will recommend, if you get it online or could buy it, read this book. This is a very interesting book which will inspire you why this kind of research is essential. It's titled as The Hot Zone, as you could see, by Richard Preston. This will give you a flair of how a disease or a pathogen travels through airways, especially through airplanes, through other means from one part of the world to the other part of the world, and if there is even slight negligence in quarantine, or if the person or the carrier of disease or the pathogen is not at a stage where you can detect that it can easily pass through the screen of another nation, and there it could spread infection. So, it is almost like a human bomb which is carrying in its own body a truckload of pathogens which can infect a wide range of population. So, this book will give you an overhaul and idea of how Ebola, one of the very dreaded pathogen, escaped from the jungles of Africa and get into the mainstream. And as we will talk about Ebola, you will realize how many epidemics it has caused and it has been causing with more and more development in the African continent. So, if we look at the Ebola virus, so there is one small aspect what I wanted to highlight in the slide. There is a renaming of the genera Ebola and the Marburg virus to Ortho-Ebola virus and Ortho-Marburg virus. So, this is the renaming which has been done. For those of you who follow this subject, just be careful, because this is a recent development which happened last year. So, keep it in mind. So, this is nucleic acid structure of Ebola. So, what Ebola is all about. So, now if we go through the slide, you will realize. So, first of all, this name Ebola, it's a very awkward name, right? This name has come from the river that surrounding where Ebola has been seen. So, there is a Ebola river, and since it was detected in that region, so it has been named as Ebola virus. Okay. So, there is no other scientific basis behind this common name called Ebola. So, if we look at the slide, the Ebola, or how they pronounce it as EBOV, was first identified in 1976. So, think of it, it is almost now 50 years when two coinciding outbreaks transpired in Sudan and in Democratic Republic of Congo DRC near the Ebola river, from which the virus takes its name. Ebola belongs to the family of Filoviridae, and I have told you the changes which has happened, and a group of single stranded RNA virus. So, when I showed you in the previous slide that, so this is how it looks like. I haven't shown the scale, and I will leave the scale for you guys to look at. Go online and check what is the scale of this particular image. If you give a Ebola search, you will get it, and that will kind of give you an idea at what scale we are operating. So, coming back to the next point, Ebola virus is primarily found in animal reservoirs. Several species of animal have been recognized as potential hosts. The natural reservoir of virus is believed to be the fruit bats. So, these bats are very common in tropical rainforest, because bats are the major carrier or operator of pollination, especially during the night. They move from one flower to another, and these are mostly the vegetarian bats which live on plant materials. So, they hop from one flower to another and they proceed with the pollination process. So, there are large number of species of bats, and if you go to a bat cave, you will realize how difficult it is to study these ones. Especially, I remember a few years back, I was in a bat cave in the mountains surrounding Madurai. They have very good bat research program at some point, and it's a very interesting experience to see these bats. So, now we are talking about the bats which are deep inside the jungle and who are involved in lot of pollination work. So, coming back to the slide, it displays a broad tendency to different cells and tissues. However, the early targets appear to be mononuclear phagocytes such as monocyte, macrophages and dendritic cell. So, this is of tremendous importance. So, in other words, this virus influences your immune system. All those cells, dendritic cells, mononuclear phagocytes, macrophages, monocytes, these are all immune cells. So, the first thing which gets compromised or which is being attacked is the immune cell of your body. The presence of Ebola virus in these organs can cause range of symptoms including liver damage, lymphocyte depletion and adrenal insufficiency. And currently, there are two main methods to diagnose EBV. One we will talk about serological method, the other one is the molecular test. Further, rapid diagnostic tests RTDs can be used for Ebola detection, and there are currently two antiviral drugs. You must have heard about these drugs. Well, we have talked about or we have learnt about the time of COVID, remdesivir and suvimab. So, these are the major broad antiviral agents which are currently available and approved by WHO. So, coming to from the discovery, if you look at it, look at the slide. So, this is the bat which is the carrier. 1976, the first outbreak in Sudan, 151 people passed away, and Democratic Republic of Congo 280. Again 1977, followed with a 2 years gap in Sudan in 1979. Then there was a huge gap. Then it again reappeared in 1994 in Gabon. 1995 again Democratic Republic of Congo. 96, look at the shift from somewhere out here, somewhere we are talking about Sudan, we are talking about Congo. It traveled all the way to South Africa. This was quite a travel. South Africa, Gabon, 2000, 2001, it moved to Uganda. Okay. So, if you look at the way it is moving, then 2001 again Gabon, Democratic Republic of Congo, Sudan, DRC, Uganda, DRC, Uganda, DRC, so on and so forth. And it has been seen till 2014, at least this report, West Africa, which is, look at the amount of travel it has done. Mali, Nigeria. So, if you look at the map of Africa, you realize that how it has spread from this corner to this, to all these areas. And now, if there is a transcontinental flying and carrying a carrier, then it can go to the other continent, and it has been seen, it has even reached Germany and other places at different times. They have been contained, but there is always a fear that it may cross to the other continent. Though, let me highlight again, they remain confined into very small pockets at different places of the world. So, this is a very interesting thing. This brings you back to the very first week when I was telling you that there is a co-evolution. Why? In order to understand biosecurity, you have to understand the evolutionary concept is there is always a co-evolution. at a particular, say, in a specific area of Himalayan belt or a specific area somewhere in the Western Ghats, there will be a unique mixture or unique number and types of organisms surviving, from virus all the way to big animals. Now, that balance can only be disturbed. Either there is a migration, which is a natural migration, as most of you know that the elephants migrate along the border of Karnataka, Tamil Nadu, Kerala. There is a huge amount of movement of animals, but that is a natural process from season to season. Similarly, there are migratory birds which are flying all the way from Siberia to India and many other places of the world where the weather is far more warmer and conducive to their survival. Now, these are natural migrations and the system is kind of adapted to it. But if there are migrations, forced migration, there are some kind of, say for example, you are introducing something, then there is a problem. And much of these pathogen spread is something to do with those kind of migration or movements of human. Okay. So, moving to the mode of transmission. So, I have highlighted they are zoonotic pathogens. They can spread rapidly among human and non-human primates. Ebola is primarily found in animal reservoirs, as I mentioned, and several species of animal have been recognized as potential host. Natural reservoir of the virus is believed to be fruit bats, and there are specific fruit bats which does carry, and more we are realizing this bat species, there is almost a huge family of bats and that number is increasing, who are carrier of different kind of virus which have the ability to infect human. So, these bats are believed to be asymptomatic carrier of the virus and can shed the virus to other animals and humans. Additionally, antibodies targeting the antigen of Ebola virus have been identified in multiple bat species. So, you see there is a very peculiar way nature has developed its immune system, and when we say that things co-evolved in a place, that means it is kind of a peaceful coexistence that you have immunity towards it, yet you survive together. So, it is kind of going hand in hand, you survive, I survive likewise. But the problem comes that if any of these elements gets either are being removed from the system or there is a new species which enter into the system by some means or other, or you take one of them and put it in another ecosystem. So, this is pretty much, if you look at it, that among the non-human primates, chimpanzees, gorillas and monkeys can become infected with Ebola virus through contact and infected bats and the carcasses of other infected animals. So, this brings us to another important point when you have to eliminate the carcass or the cadaver, you have to do it in a very confined environment. So, if we look at the routes of Ebola virus transmission, so here is the virus, direct and indirect fluids, animal transmission, sexual contact, mother to child transmission and airborne transmission. So, if you look at it, there are multiple ways it can get transmitted. So, that is why I was telling you about the airborne transmission. I was telling you, if a carrier moves to another country and sneezes, coughs or something, you are going to transmit this virus. And this is a much more pictorial demonstration of the Ebola virus ecology and transmission for your better understanding. Animal to animal transfer. Evidences suggest that the bats are the reservoir of host of Ebola virus, and bat carrying the Ebola virus can spread the virus to other animals, as we have already mentioned. There is a spillover event. A spillover event occurs when bat, ape, monkey or person become infected with the Ebola virus through contact with the reservoir host. human to human transmission. This could happen through any kind of fluid transfer or even mother to child transfer. Then comes if there are people who can withstand, it means they have inbuilt immunity within their system. And areas where one has to be careful is how a person or an animal passes away because of Ebola virus, how to eliminate. And this is of extreme importance, and healthcare professionals have to be extremely careful during this situation, and unprotected contact of blood and other body fluids. These are of immense significance in order to control these kind of virus infection. Now, if you look at the Ebola virus genome, this is pretty interesting. The genome of Ebola virus is nearly 19 kilobase in length and encodes 7 structural proteins. Nucleoproteins NP, RNA-dependent RNA polymerase, which helps in the replication, RNA-pol, and nucleoprotein carrying some of the critical information, virion protein VP30, VP40, GP, VP30 and VP24. These proteins form virus particles and play essential role in viral replication, transcription and pathogenesis. So, what is the significance of these different kind of particles? These particles or the proteins they encode or the molecules they encode, they are extremely critical about how the virus anchors onto the cell, how it enters inside the cell, how it replicates inside the cell. And while telling this, you have to realize this is all being carried out by a fragment of RNA. So, it has a specific sequence which does a specific set of jobs. So, if you understand the specific area of the RNA, what it is doing, you can always raise antibodies or something which will block that. So, that is where the whole area of discovering antidotes starts. And second thing, this sequence of RNA, in this case of Ebola, is extremely important because you can use this sequence for all sorts of detection devices, because this is the key and this is the basic of understanding and all those proteins which they are producing. So, you can raise antibodies against them, and that is how most of the serological testings are being done. So, in terms of what all it affects, muscle pain, hemorrhage, and this is very, very critical, chronic rheumatological inflammations, adrenal insufficiency, this is another key thing, arthritis, encephalitis, tremendous fatigue, sore throat, lymphocyte depletion. This happens because of it attacks the immune system, liver damage, these are critical ones, and bleeding gums and nose and vomiting. These are the ones which cause the maximum amount of human fatality, because when you talk about breakage in the blood vessels, then it is extremely challenging to revive the patient. So, these are the most critical symptoms which you see when infected with the Ebola virus. Generally, there are two main methods used to diagnose Ebola: the serological testing that detects the presence of antibodies against EBOVs, the Ebola virus, in a patient sample using ELISA or IFAT. Molecular tests like RT-PCR and LAMP, most of you must have heard about RT-PCR during the time of COVID. There is a rapid test called RE-EBOV, antigen rapid test. So, these are mostly tests which relies on this structure. All the serological testings which are being done purely relies on the structure of the nucleus or nucleic material. In this case, it is RNA and the proteins encoded by those RNA. So, that is the key for all the serological test materials which are being developed. The other sort of sensors what we will be discussing in this course, which is the most economical one and does not need extreme sophisticated instrumentation and is very rapid and fast, are electrochemical sensors. And we will talk as we will progress. By the next week, we will definitely discuss some of these sensors which have been developed against Ebola as electrochemical sensors. And this is the area which we are going to deal for next 5 to 6 weeks, where we will talk about all the different kind of electrochemical sensors which are currently available and what are their advantages, disadvantages, where next technological breakthroughs are to be made, and how they could be miniaturized and how they could be made available to a large population. Okay. Now, in terms of the Marburg, which is pretty similar to the Ebola and almost in the same family, these are the years when you have seen it's in the reverse order, when we have seen they have appeared time and again. So, starting from 1967 in Germany, Yugoslavia, it's no more, it has now broken down into different other countries, South Africa, Kenya, DRC, Angola. So, if you look at it, United States, again there are carriers, Netherlands, and the last documented is 2017. I am pretty sure there are other spikes which have happened before this, but this is from the WHO report. I am quoting for you. These are the cases of Marburg. And again, in the same line like Ebola, there are detection systems, but it's still a lot more to be done in terms of understanding how Marburg has spread over the years. So, if you look at Marburg, formerly known as Marburg hemorrhagic fever, is a severe, often fatal illness in human. The virus causes severe viral hemorrhage. As I mentioned you, the major area which it gets affected is the blood vessels. It is out here. The most of the bleeding, which is the major cause of concern when a person gets affected with Marburg, and same as Ebola. The average MVD case fatality rate is around 50%. Early supportive care with rehydration, symptomatic treatment improves survival. There is also yet no licensed treatment proven to neutralize the virus, but a range of blood products, immune therapies and drug therapies are currently under development. And the fruit bat are considered to be the natural host, same as Ebola, of Marburg virus. The Marburg virus is transmitted to people from the bats and spreads among humans through human to human transmission. Community engagement is the key for successfully controlling the outbreaks. So, if you look at both Marburg and Ebola, they both carry a very similar pattern of disease transmission, and both of them in nature co-evolved in specific areas. Now, if you isolate from their specific area and bring them to another area, they act like novel weapon hypothesis, exactly following what we have discussed in the first week, the novel weapon hypothesis. They become a novel weapon because why they become a novel weapon in terms of chemical aspect, if you look at it. Now, they are producing those kind of proteins which are not being seen by the new population where they enter, totally alien. The body has no idea what they are producing, and the very moment they get an entry, they could completely alter the system because body is not prepared to have a Ebola antibody or a Marburg antibody in their own body. So, they could gain access into our body very fast as compared to a virus which is coexisting in our system. So, look at it from any of these things from the evolutionary perspective. So, when you study about COVID, you think of it, how it has evolved over the years. COVID is nothing new. COVID was there for a long time, but it's just spiked up at a particular time and completely crippled the global economy. So, most of these things are there, and a lot more research has to be initiated to have our protective arsenal ready for any kind of such a spike, because we cannot stop movement. So, we have to be practical. There will be movement of human being from one place to another. There will be transmission of goods from one place to another. There will be a trades and international mixing. What can be done is that to develop antidotes, to develop therapies, to develop medications, the very moment such things spikes in any part of the world, and that also demands a large amount of global cooperation. Thank you.